More second messengers. There's really one left to talk about, one second messenger system left to talk about, which is diacylglycerol and IP3, inositol triphosphate. I know that sounds like a mouthful, but it's not so bad. So, I think this is the third system, right? We've done calcium as a second messenger, and we've talked about the cyclic AMP or cyclic GMP, right? Cyclic nucleotides as second messengers. We'll talk about DAG and IP3 now. Diacylglycerol is DAG. Inositol triphosphate is IP3. And the way that these messengers get liberated is through activation of an enzyme, PLC. Okay, and so potentially, right, a G protein coupled receptor is activated, and the enzyme that it targets to turn on is called phospholipase C. So activation of an enzyme PLC, phospholipase, and it might be worth writing down because it tells you a little bit about what it does, not just its structure. So that's the enzyme, and it's a lipase. Not just that, it's a phospholipase. So it cleaves membrane phospholipids. And so, again, where does this get turned on? It may get turned on by a G protein. Okay, it's a very common way that this enzyme is activated. When it's activated, it's able to cleave a phospholipid that's in the membrane called PIP2. Right, so this is a membrane phospholipid. Phosphatidylinositol phosphate 2. Don't need to know the big ugly name, but PIP2, right, is a membrane phospholipid. PL phospholipid is cleaved by this enzyme. And there are two products, DAG, diacylglycerol, and IP3. Okay. Each one has a particular target. The remainder of the PIP2, the PIP2, that's the diacylglycerol. This portion sticks in the membrane. Okay, so DAG remains in the membrane. Whereas IP3 is liberated from the membrane. It can signal within the cell. So released, into the cell. DAG can go on to activate protein kinases, like protein kinase C, something that we'll talk about momentarily. So we'll talk a lot about kinases in general and a couple of specific kinases. Whereas IP3 can activate IP3 receptors. These IP3 receptors often serve as gates for calcium to leave places like the ER. So activation of IP3 receptors often triggers calcium release. And we've talked a lot about calcium already. Okay, and so you can see on the figure here, right? Here's the PIP2 that's in the membrane, made up of sort of two parts, right? The diacylglycerol and the IP3. And nothing's going to happen with it until this enzyme, PLC, phospholipase C, can activate it. Once it does, right, there are two products. It basically makes a cut, sort of in half, if you will. So it cuts this. Some part of that initial PIP2 is left in the membrane as DAG, okay? And some part is liberated, it's free, IP3. And you can see IP3 then works as a ligand to activate its receptor right here. So if we jump back to the picture that's shown on the left, here's IP3 receptor, here's the IP3. So now what this does is it opens up the calcium channel that's within the cell, right? This is not a calcium channel that's on the membrane surface, but this is a calcium channel that's on the ER, and this leads to an efflux, a release of calcium from the intracellular stores, okay? So basically this is just a chain reaction. You can go all the way back to activation of a G protein coupled receptor and work through this DAG IP3 second messenger pathway to get to a calcium release, and then calcium will have multiple downstream targets, okay? So I think, we've gone through these 3 second messenger systems. Let's talk about some of those targets. But first, we need to talk about second messengers before we talk about their targets, right? The way that second messengers work, I think this is a really important point. Second messengers regulate phosphorylation state. The phosphorylation state of intracellular proteins, okay, by adding or removing a particular phosphate group, and that phosphate group comes from ATP. So again, the question goes back to, well, how does ATP provide cellular energy? Well, one of the ways that it does this is that one of those phosphates from ATP can be liberated and transferred to a molecule that may need it in a reaction that requires energy input. Okay. So shorthand, right? If I want to draw that something is phosphorylated, I often just draw a phosphate group attached to it. So now I've phosphorylated the word phosphorylation. Okay, so that's kind of my shorthand. This is done by kinases, right? Kinases phosphorylate. It's ugly. Oh no. What happened? Okay, so what was I saying? Kinases phosphorylate. So these are enzymes, right? Proteins that catalyze chemical reactions. Kinases do the phosphorylating. Whereas protein phosphatases dephosphorylate. So you can think of this generally as activation, inactivation, phosphorylation, dephosphorylation, kinase phosphatase. So kinases dephosphorylate. Now typically, protein, as you know, is made of a chain of amino acids, and often the kinases and phosphatases will both act on serine or threonine residues. Right, so these are a couple of amino acids, serine, threonine. And so in the long amino acid sequence of a protein, it's not that a phosphate group is attached, to the entire thing. It's got to be attached at a particular spot, and those residues are the ones that are targets for phosphorylation. There are also a family of kinases, right? I mentioned these before, RTKs, receptor tyrosine kinases. And these are sort of outside, now they're kinases, right? But they're not the ones that are going to move within the cell to target. These are membrane receptors, okay, that are able to phosphorylate when activated by an extracellular signal. Okay, so I think we talked about this in one of our previous slides, right? That there's a way that the ligand can transduce to activate an enzyme directly, right? One that's stuck in the membrane. So what we can say is that they're activated by extracellular signals. There's receptors activated by extracellular, EC, extracellular signals, right? Ligands. And they phosphorylate. The T is tyrosine. Okay, so R receptor, T tyrosine, K kinases. So they're sort of a special category of kinases that aren't sort of free in the cell. These kinases are bound to the cell membrane, right, where they'll be activated. So let's take a look at the figure above, because I think this is a pretty concise way of stating what we're trying to state. So Second messengers are working on this kinase here, right? So some of the second messengers that we talked about, cyclic AMP or cyclic GMP, potentially calcium that's activated with calmodulin and complex, right? Maybe IP3 or DAG serve as a second messenger to activate this kinase. This kinase is now on, right? So it's going to work. And what it's able to do is take a protein in its sort of inactive form here and attach this phosphate group. So you can see that this protein here, where the arrow is, has been phosphorylated by this kinase. Okay, now it's going to be active, right? Now it's going to be able to target multiple other things within a cell, multiple pathways. And I'll show you examples, a lot of examples of what phosphorylated proteins can do. And this will be active until a phosphatase is able to dephosphorylate, that is, remove this phosphate group. And now we have our protein here in our inactive state. Again, importantly, where does this phosphate group come from in the first place, right? It's the job of the kinase to transfer one of those phosphate groups from ATP, right, eventually to the protein target. Right? The protein target can't get phosphorylated on its own. It requires a kinase to do that. Okay? So we're going to go through a bunch of different kinases, right, that are often targets of second messengers. The first kinase that we'll mention is protein kinase A. All right. I think I never want to lose sight of what activates it. We're going to talk about its structure and how it works. But protein kinase A is typically activated by cyclic AMP. So activated. Right, so we have a second messenger that's regulating a phosphate. That's kind of the theme of what we're going to talk about. pKa has 4 subunits. So again, it's a protein, 4 subunits. There are 2 domains that are catalytic. And then there are 2 domains that are regulatory. So it's at the catalytic domains where phosphate groups will be transferred. Okay. The cyclic AMP is able to bind the regulatory subunit. Okay. And this has an effect of releasing the catalytic subunits. Okay, so normally the regulatory subunits are bound and the protein kinase is inhibited, but when pKa is present, excuse me, when cyclic AMP is present, let me back that up, now the catalytic subunits are free. Okay. Again, this kinase will go on to phosphorylate serine and threonine residues of targets, right? So pKa, right, can then transfer, so a protein that's inactive, so we'll say that it's off. The same protein is now on when it has a phosphate group attached to its serine or threonine residue, right? And so I think the picture is fairly helpful. Here's pKa, right? Roughly what it looks like. There are different domains. You can see that the catalytic subunits here and here are sort of docked, right? And then the regulatory domains have spots for binding, right? So when cyclic AMP is present, it will bind at these 4 different, that sort of look like invaginations in the protein here in the sketch. And so with pKa off, nothing happens, but when cyclic AMP is present, so cyclic AMP is bound here, and here, and here, and here, and this frees pKa to go phosphorylate targets.